So all the cellular material between the plasma membrane and the nucleus is in what we call cytoplasm. That cytoplasm is composed of many things, most of which we can't see with a light microscope. It would include cytosol, which would be the gel-like solution of mostly water molecules. Inclusions that would be insoluble molecules. Those would change with different cell types, glycogen granules and some pigments and others. Organelles are the metabolic machines that run the cell. Some of them are surrounded by membranes and others do not have membranes. And those organelles are going to be the focus of this. Those that are surrounded by membranes include the mitochondria, the endoplasmic reticulum, the Golgi apparatus, peroxisomes, and lysosomes. Those without membranes would be the ribosomes, the cytoskeleton, although the cytoskeleton would be enclosed by the plasma membrane, and the centrioles. The fact that they have membranes is very important because if you have a membrane, you can separate the environment inside that membrane from the environment outside, and you can do things that you normally couldn't do. So mitochondria are called the powerhouse of the cell. They produce most of the cell's energy molecules, ATP. They do that through aerobic, which is oxygen requiring cell respiration. The Krebs cycle, electron transport, all take place within mitochondria. We'll talk more about those when we talk about powering muscles later on. They have a double membrane, and their inner membrane has many folds called cristae. Within those cristae are the proteins used for electron transport, as well as some of the Krebs cycle enzyme. Mitochondria have their own DNA with a slightly different genetic code, their own RNAs, and their own ribosomes. They resemble bacteria. They are capable of the same kind of cell division bacteria use called fission. In fact, the working theory is that mitochondria are parasitic bacteria that got into a cell and took hold. Ribosomes do not have a membrane around them. They are the site of protein synthesis. They are made up of both protein and ribosomal RNA. They are found freely in the cytoplasm, making proteins there. Many of them are also bound to the endoplasmic reticulum, where they are building proteins into the endoplasmic reticulum. And that endoplasmic reticulum is a series of interconnected cisterns or tubes that enclose fluid-filled interiors. The endoplasmic reticulum is continuous with the outer part of the nuclear membrane. And as we talked on Tuesday, there are two kinds of endoplasmic reticulum: rough endoplasmic reticulum and smooth. The rough endoplasmic reticulum looks rough because it's studded with ribosomes. Those ribosomes are synthesizing proteins. Those proteins are being built into the internal part of the endoplasmic reticulum, where they are modified and folded correctly. And they are then sent to the Golgi for further processing and delivery to where they are going. The smooth ER is found is used for lipid metabolism, things like cholesterol and steroid-based hormones. It's also important for absorption, synthesis, and transport of fats. There are detoxification enzymes for certain chemicals, and it's also where glycogen is converted to free glucose. And it is a modified form in muscles called sarcoplasmic reticulum that is used for the storage of calcium. From the endoplasmic reticulum, materials move to the Golgi apparatus, which modifies, concentrates, and packages those proteins and lipids from the ER. So we need transport vesicles from the ER to fuse with the inner or cis face of the Golgi. proteins or lipids are then taken inside, modified, tagged, sorted, packaged. So the Golgi is then the kind of the traffic director. Packaging and distribution center is the analogy that I like to use. It controls which of the three final pathways the products will take to go out the outer face, which is the trans face of the Golgi. So pathway A is for secretory vessels. Those are proteins that can be used outside the cell. They will fuse with the plasma membrane and undergo exocytosis, as we talked about earlier. Pathway B, the vesicles contain lipids or transmembrane proteins. They're also going to fuse with the plasma membrane or the membrane of some organelle and put their contents directly into that membrane. And pathway C is lysosomes that contain digestive enzymes remain in the cell, and they hold the contents in the vesicle until needed. So if we look again at the sequence of events from protein synthesis on the rough ER to the final distribution, it starts with proteins being built into the rough endoplasmic reticulum. When they reach the end of the endoplasmic reticulum, they are packaged in a vesicle that goes off the trans side of the endoplasmic reticulum and merges with the cis side of the Golgi apparatus. They then move through the Golgi apparatus where they are concentrated, modified, just put into whichever pathway they are going in, either A, B, or C, as we just discussed. Another internal structure is the peroxisome, which is a membrane sac containing detoxifying substances such as free radicals. Those free radicals are toxic, highly reactive molecules, and peroxisomes can get rid of those. The two main detoxifiers are both oxidases, which use oxygen to convert toxins to hydrogen peroxide, and then the hydrogen peroxide is converted to water by an enzyme called catalase. So oxidase and catalase are the two main enzymes of peroxisomes. They do also have a role in the breakdown of fatty acids. Lysosomes are spherical membranous bags that have digestive enzymes. These enzymes are acid hydrolases. They're inside the lysosome, so they don't damage the rest of the cell. They're involved in digesting ingested bacteria, viruses, and toxins. They will also degrade non-functional organelles. They metabolically are used to break down and release glycogen and release calcium from bone when that's necessary. And if they are opened up inside the cell, they can cause the cell to digest itself, which is a process known as autolysis. So lysosomal storage disease results when one or more lysosomal digestive enzymes are mutated and do not work properly. A very famous lysosomal storage disease is Tay-Sachs disease. There's an enzyme missing to break down glycolipids in brain cells. Therefore, the glycolipids build up, interfering with the nervous system. It's seen predominantly in infants of Central European Jewish descent. It does cause seizures, mental retardation, blindness, and death before age 5. But you should also be aware that there is a similar risk factor for French Canadian descent people. They carry the mutation at about the same rate as the Ashkenazi Jews. So the endomembrane system is the organelles we've discussed so far, really the ER, the Golgi apparatus, secretory vesicles, and lysosome. We would put the nuclear and plasma membranes in there as well. That's the internal membrane system. Those are going to work together to produce, degrade, store, and export biological molecules, get rid of potentially harmful substances.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
The cytoskeleton is a network of rods that run through the cytosol. There are hundreds of different proteins involved in the structure, but the cytoskeleton is going to act like your bones, your ligaments, and your muscles, playing a role in the movement of cells and components within the cells. The three major types of fibers we'll talk about are microfilaments, intermediate filaments, and microtubules. The microfilaments are the thinnest of all the cytoskeletal elements. They are semi-flexible strands of a protein we'll see a lot this year called actin. Each cell has its own unique arrangement of those strands, and they share what's known as a terminal web, which is a dense cross-linked network of microfilaments attached to the cytoplasmic side of the plasma membrane, which strengthens the cell and helps it to resist compression. Some are involved in cell motility, changes in cell shape, endo and exocytosis. Here's a cell stained for microfilaments. Intermediate filaments are between microfilaments and microtubules. They are tougher, more insoluble. They are rope-like protein fibers. They're composed of a tetramer, which is 4 fibrils twisted together, giving you a strong fiber. These are very good at resisting pulling forces. They're going to attach to desmosome plaques and act as internal guy wires, for example. And some of them have special names. So in the neurons, we'll call them neurofilaments. And in epithelial cells, we'll call them keratin filaments. Microtubules are the largest cytoskeletal elements. They consist of hollow tubes composed of protein subunits made up of tubulin. They are constantly being assembled and disassembled. They radiate from the centrosome area of the cell. They determine the overall cell shape and distribution of organelles. A lot of organelles are tethered to microtubules to keep them in place, and many substances move throughout the cells along these microtubule tracks using motor proteins. Those motor proteins are little machines that do what you would expect them to do. They move things from place to place. They carry organelles from one part of the cell to another. They use microtubules as their tracks, and like most things in the cell, they are powered by our energy source, ATP. The centrosome and centrioles. The centrosome is located near the nucleus, which means cell center. It's a microtubule organizing center consisting of a granular matrix and centrioles, which are a pair of barrel-shaped microtubular organelles at right angles to each other. The newly assembled microtubules radiate out from the centrosome to the rest of the cell. Some microtubules also aid in cell division, we'll talk about a little later. And some form part of that cytoskeletal tract system. They are also going to serve as the basis for 2 membrane extensions we'll talk about, cilia and flagella. Those cilia and flagella aid in the movement of the cell or materials across the cell, as well as microvilli, which are going to be a little smaller than cilia, we'll look at. Cilia are whip-like modal extensions on the surface of a cell. Respiratory cells are a good example. And they work together by beating in a single coordinated direction to expel substances across a surface. Mucus is moved up our respiratory tract by these cilia. Flagella are long extensions, and they are used for cell motility. Again, only sperm cells are the only flagellated cell in humans. The only one we'll talk. They're both cilia and flagella made up of microtubule structures that are synthesized by the centriole, called basal bodies, and they form the base of each of those. Cilia and flagella that have a 9+2 pattern of microtubules, so 9 double tubes surrounding a central pair of doublets. And cilia movements, again, alternate between a power stroke and a recovery stroke, which gives you a current that moves things across the cell. As shown here.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Microvilli are even shorter. They're finger-like extension of the plasma membrane projecting from the surface of certain cells, and that increased projections are going to increase the amount of surface area that's available for absorption. Microvilli have a core of actin filaments that's used for stiffening the projections. And that will bring us to the nucleus, which is our next section.